So my senior design project is on creating and developing EPIC, a tool for gene regulatory network reconstruction. Gene regulatory networks, or GRNs, are essentially just a set of interactions between genes, transcription factors, and other protein regulators that control genetic expression and cell function. We can visualize these networks with diagrams like the one I have on the left. They show the interactions between the genes and transcription factors. This can be particularly helpful within the field of cell plate engineering, where researchers perturb the expression of different genes in cells in order to convert cells from one type to another. Reconstructing networks and consequently identifying the different interactions can help researchers take a more targeted approach in cell plate engineering. If they know exactly which interactions take place, they can have a better idea of which genes and transcription factors will be best to up or down regulate to get their desired results in cell plate engineering. Therefore, our need statement would be as follows. Cell plate engineering researchers need a convenient way to quickly and efficaciously identify gene and transcription factor interactions in different cell types in order to increase the success in cellular reprogramming experiments. So we can reconstruct these networks using genetic expression data. Currently, there are computational tools out there that can take in this genetic expression data, and from that data, guess which interactions take place in the network for that cell type. However, many of the tools are problematic because they're computationally burdensome and output networks with a lot of false edges. This brings us to EPIC, a tool in R that was developed in the Kahan lab that utilizes single-cell expression data in order to reconstruct GRNs. EPIC builds off of another algorithm that reconstructs GRNs called CLR, which utilizes mutual information between gene pairs to reconstruct the network, but it adds another additional step to increase the precision and speed of reconstruction. When EPIC was benchmarked against other existing algorithms, it showed a rate of precision almost twice as great. Therefore, we believe that this tool shows much promise, but we would still like to increase the overall speed and user-friendly nature of this tool to make it more appealing to researchers. This now brings us to my project. The goal of my project is to take the EPIC algorithm that we have in R and translate and develop it in Python. There are two main reasons why we believe having EPIC in Python will be advantageous over R. First, we believe that having a tool in Python will increase the speed and the computational power, potentially speeding up some functions in the tool. Next, Python will also add to the convenience of the tool, since we can make it compatible with the Python package Scampi, a tool that many researchers utilize to work with single-cell data. Scampi is used for data preprocessing, so if EPIC is compatible with it, then processing of the data and the reconstruction of the network can be done in one setting, which will likely be more convenient to the researcher. In addition to these two improvements, we also hope to further develop the tool in Python with the hopes of even further improving the speed and precision of reconstruction, as well as the user-friendly nature of the tool. Now for the current results I have so far. I currently have the original EPIC in R, as well as the translated version in Python. The first thing I wanted to check with my translated version of EPIC in Python was the performance. I wanted to ensure that EPIC in Python performs with just as much precision as EPIC in R. To do this, I reconstructed the network from a muscle data set that has the gold standard reconstruction, so we can measure the precision in reconstruction with both tools. As we can see through the precision recall curve, as well as the AUPR reconstruction, EPIC in Python performs essentially identically to EPIC in R. A higher AUPR refers to a greater precision in reconstruction, and both versions of EPIC have a higher AUPR than other tools for GRN reconstruction, such as CLR or GA3. Therefore, I've likely met my goals for precision with this EPIC in Python. And some next steps to this project will include getting feedback from the beta testers for this tool on the efficiency of the code and the user-friendliness of the tool, as well as integrating the outputs of the EPIC in Python with the AND data structure in Scampi, instead of storing the network in data frames. And this incorporation of the AND data structure that in the Scampi package can add to the convenience for the researchers. And that's what I have for my project. And thank you so much for listening.